As we prepare for Pakistan's national anthem, I kindly request everyone to please rise on their seats. When we begin the session, may I invite Dr. Muhammad Islam for recitation of the verses from Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم والتين والزيتون وطور سينين وهذا البلد الأمين لقد خلقنا الإنسان في أحسن تقويم ثم رددناه أسفل سافلين إلا الذين آمنوا وعملوا الصالحات فلهم أجر غير ممنون فما يكبين صدق الله العظيم.
Honorable Dean, Faculty of Life Sciences, Professor Dr. Fahim Aftab Sahab, Professor Dr. Mazur Rahman, respected faculty members, dignities, colleagues and participants, Assalamu alaikum. I, Dr. Nila Yasmin, welcome you all in the inaugural session of Cell Culture Techniques for Mammalian Cells. To begin with, please allow me to introduce and invite our esteemed dignities on stage. To start with, let me introduce the Dean Life Sciences, Professor Dr. Fahim Aftab Sahab. He is a distinguished academic and researcher, currently serving as a full professor, ex-director at Institute of Botany and Dean Faculty of Life Sciences, University of the Punjab, Lahore. With an MSc from Punjab University in 1990 and PhD in 1997, he specializes in protoplast culture technology. Dr. Aftab has made significant contributions to the field of plant biotechnology and regenerative biology. His extensive research background, including fellowships in the USA and Germany. He has also run several research projects, including the ones from Higher Education Commission of Pakistan and the Alexander von Humboldt Foundation, Germany. His current interests focus on woody plant propagation, endophytic bacterial communities, and proteomics. Please give a big hand and let me invite Professor Dr. Fahim Aftab Sahab on stage. So please come to stage and share the session. By the way, this was a small hand. Please give a big hand to the Professor Aftab. It's time to introduce our esteemed director, Professor Dr. Mazur Rahman. I'm honored to extend an invitation to Professor Dr. Mazur Rahman, a prominent figure in the field of structural biology, vaccine development, and disease diagnostic. Professor Rahman's extensive academic journey, marked by significant contributions to the understanding of membrane proteins and viral proteins, structure-function relationship, underscores his invaluable expertise. With a steadfast commitment to advance scientific knowledge and fostering collaboration, he has been a very well manager and has contributed a lot during his tenure at CEMB. Please welcome the patron of the workshop and director of CEMB to come on the stage and join the share. So please come on the stage. It's time to introduce the focal persons of the workshop. Above both of them is obviously Professor Dr. Muhammad Idris Saab, who is a distinguished molecular biologist and virologist with over 20 years of academic research and administrative experience. He completed his PhD in molecular biology and postdoctoral research at CDC in Atlanta. He established a PCR-based diagnostic lab in Pakistan, providing affordable diagnostic services and developing low-cost kits for diseases like hepatitis, tuberculosis, and COVID-19. With over 200 research publications and numerous national and international awards, including the Tawaz Fellowship and PES Gold Medal, Dr. Idris is leading expert in his field. He has also served as the Vice Chancellor of Hazara University and University of Peshawar. He continues to contribute to molecular biology research and education. Please give a big hand to Dr. Muhammad Ali. Sir, please come forward. And our second forward person is Dr. Shazia Rafiq, who is serving as an associate professor at CEMB. She has done her PhD from biology lab in 2011 from the same center, focusing on the generation of infectious hepatitis C, pseudoparticles, and their neutralization assays. During her PhD, she was awarded multiple prestigious travel grants from institutions in Korea, India, and USA. She has also done postdoc fellowship from LUMS, School of Biological Sciences, and in 2014, she was selected for a fully sponsored course on protein purification at Cold Spring Harbors Labs, USA. Her current work includes the epigenetic regulation of PDE4 gene in glucose metabolism. Dr. Rafiq's research interest spans biology, epigenetics, HCV-induced type 2 diabetes, and therapeutic vaccines. Please give a big hand to Dr. Shazia Rafiq. Now may I request Professor Dr. Muhammad Ali Sahab to please come on stage and give an introduction of the program.
नहमदुहु व नसल्ली अला रसूलिल करीम अम्मा बाद फ़अऊज़ु बिल्लाहि मिनश शैतानिर रजीम बिस्मिल्लाहिर रहमानिर रहीम. Honorable Chief Guest Professor Dr. Fahim, Dean Faculty of Life Sciences, Professor Dr. Mauzur Rahman, Director CMB, Professor Dr. Kausar, Meritorious Professor, faculty members, dear participants from different universities and institutions, Assalamu alaikum and a very good morning. It is a matter of immense pleasure for me to welcome you all to this workshop on mammalian cell culture techniques. Whether you are a seasoned researcher and emerging scientists or curious students, this workshop offers a valuable opportunity to deepen your understanding of this fundamental area of biotechnology. Mammalian cell culture plays a pivotal role in various fields, from biopharmaceutical production to genetic engineering and cancer research. The techniques we will explore together are the building blocks for the advancement in the medical treatment, vaccines development, and understanding complex biological process, inshallah. Over the next two days, we will be delving into the essential aspects of mammalian cell culture, including aseptic techniques, media preparation, cell seeding, cell passaging, the cell viability transfection, cell counting, the cell line maintenance, and troubleshoot common issues. Our expert instructor scientists that we have three today, they will share their insights and experiences, and I encourage you actively participate in the hands-on sessions. And also, this workshop is not only about gaining technical skills, but also fostering collaboration and exchange of ideas. So please take the work with your peers and instructors and make the most of your resources available to you. Once again, I extend warm welcome and wish you all a productive and enlightening experience. Let's take this opportunity to push the boundaries of what we can achieve with mammalian cell culturing techniques. At the end, I would like to express my gratitude to our esteemed chief guest, Professor Dr. Fahim Saab, joining us today for this inauguration of this workshop on cell culture techniques. I am also thankful to our worthy director, Professor Dr. Mauzur Rahman, for his continuous support, encouragement, and guidance in making this workshop a reality. Last but not the least, on behalf of CMB family, I would like to extend our deepest thanks to the Worldwide Scientific Ion Molecular Products and Pakistan Science Foundation for their generous sponsorship to this workshop. Thank you so much.
Thank you, sir. Now Now please, may I invite Director CEME Professor Dr. Mazur Rahman Sahab to please come on stage and give a brief introduction of the activities being held at CEME. Big hand is big hand.
بسم الله الرحمن الرحيم رب اشرح لي صدري ويسر لي امري واحلل عقدة من لساني يفقهوا قولي. So first of all, on behalf of the CMB and the CMB faculty and the colleagues, I welcome the participants to this mammalian cell culture course, which of course has an enormous application in every field of life sciences. So I also take the opportunity to thank and welcome the Professor Fahim, who has been recently appointed as Dean of Life Sciences. And we always get enormous support from the University of the Punjab, although we are, I think, 7-8 kilometers away from the main campus, but their enormous support is with us. So most of the students and the participants also know, we are running the MPhil and the PhD program, which is unique to molecular biology subject. And this was only possible with the, a lot of support from the University of the Punjab. So on behalf of the CMB, I appreciate the contribution of the University of the Punjab, not only the establishment of this center, also to keep progressing this center from basic research level to the applied aspects. So coming towards the center, particularly for the participants, what we are doing in the center, basically we are doing research in two major areas. One is the agriculture and second one is the health sciences. In agriculture in Pakistan, this center has this unique position, the only and the unique cotton GMO varieties which are being cultivated throughout the Pakistan and now is getting the share up to 50 or 60% of total cotton cultivation in Pakistan are invented by this center. So this is the huge contribution that saves not only millions, I would say billions of the dollars at the doorstep. This center has been and stem cells, genetic diseases, and also the health scientists also contributed towards the development of the GMO cotton, because they are working on the BT strains and they found those toxin genes which were introduced into the cotton plant, and cotton plant become resistant to the insects. So this was a joint venture between the kind of health sciences scientists and the agriculture scientists at the Center of Excellence in Molecular Biology. And when there was a pandemic before SARS-CoV-2, I would say between 2005 and 2008, this center has enormously contributed towards the diagnostic of the HCV, when more than 12% of Pakistani population at that time was affected by the hepatitis C virus infection. And this center has the privilege to discover, or I would say to identify first time in Pakistan that genotype 3A is the most predominant genotype of HCV in Pakistan. And that was well then recognized and well popularized all over the world. So this huge contribution, because before that the one antiviral drug was being used for treating genotype 1, 2 and 3. And with this will be the approach like a personalized medicine. So we have to target the genotype 3A. The course which is being organized today is basically by the same virology group who has enormous contribution in the field of virology in Pakistan. And this is beyond some time, mind you, at that time the data was published with more than 30,000 patients sample. This is, you see, enormous human sampling data, and on that basis the results were inferred that genotype 3A is the most common one in the Pakistan. So actually this is the mandate of each and every center in its area of specialties, that if there is a pandemic, if there is any kind of the sudden the disease occur in the country or come somewhere else, then it is the duty of the research centers to help the nation to address those problems. So I'm very glad that the scientists at the CEMB not only fostering the basic research, they are also working on solution-oriented research, which is required for the Pakistan. So coming towards the stem cell research, nowadays there are so many diseases, autism, and there are some, kind of the like diabetes foot injuries that can't be repaired, in the diabetic patient. So with the use of the stem cells, the research experiments have been performed at the center where a patient having the diabetic foot injury could not be, have the healthy foot for a year. So when the stem cells were applied, so his foot was recovered within 25 days. So this is the high-tech research. And in stem cell, again, this center is at the forefront in Pakistan. And now we are in phase of clinical trials and approval with the DRAP to bring this, expensive kind of the therapeutic nature of the stem cells to the population. So just I would like to share now one therapy of the stem cells which is being imported. Sometime it costs up to the 50 lakh rupee for one patient. The center has made such kind of the stem cells which will cost up to with 10 times less cost, for example 5 lakh rupee. So this is only possible, the scientists working in local environment and taking care of the local population and to bring such kind of the high-tech therapeutic interventions and the solutions for the local population. So coming back to the mammalian cell culture, as I said earlier, in every field of life sciences, while we produce the GMography, we check the toxicity of the cell, that needs mammalian cell culture technique. While you develop a vaccine, you need to propagate the vaccine, viral vaccines, you need a mammalian cell culture techniques. While you want to study the differential epigenetics or any kind of the occurrence of the viruses, you need a mammalian cell culture technique. Even in some kind of the bacterial population, the toxin also need the mammalian cell culture techniques. So this is kind of, I would say nowadays, such kinds of techniques is like a spectrophotometer. Everyone has to know how this can be performed. As you measure the DNA concentration or the protein concentration by putting the simple keyword in a spectrophotometer, now I would say in this modern arena, in case of the life sciences, every student must have the knowledge to do how to do the mammalian or the cell culture techniques. Because with this, you can revolutionize your project. You don't need to go to the animal model. You can do in 96 well the assay for which you need 100 mice. And you can think of if you want to keep the 100 mice and then 100 rabbit, and then if you are going to develop an animal vaccine and you have to have the 100 buffalo, so what will be the cost of the experiment? And look at the 96 well plate having a variety of the cells, and you can look at the same properties and the characteristics which you look in an animal model. Of course, animal model is required at the later stage to reach to a final drug development, but at initial stage for screening purposes and for initial characterization, these are the mammalian cell culture techniques. So this is the importance of the today's course, and you are lucky have to be here, and we are lucky as well because we are having the participants from the different institutions of the Pakistan, and I see the faces of the faculty members from Islamic University, Bahawalpur, and from other institutions as well. They traveled a long way to get to the Lahore. So this will be opportunity on both sides to learn and to share the ideas with each other. And at last, I would say this opportunity, please don't take it only the mammalian cell culture, make friends at the center. And we always encourage those projects which have some applied outcome. And being a director at the institute, I believe in research grant writing. first develop the ideas, work together to some extent, and then write the research grant. Because without having the funds, nothing is possible in the world. So this is the basic criteria for every and each scientist to write the grant. And for the faculty members which are put particularly here from the other institutions, they may find some collaborative research and come together to serve this nation. So at the end, I thank you, the Fahim Saab and the course organizer, Dr. Adriz, and Dr. Shazia sitting next to me. She put a lot of effort. And we have a team of actually the players at the center, and for which we don't need to organize the committees and the other groups to organize a coursework. And this is ready to made cake, and Dr. Anila always had this kind of activity and others. And this is you, other people who came from other institutions, they can also get benefit from us. That having such course is not, we don't put a lot of effort. Our people are so trained and they work professionally and organize such kind of the course. Of course, the co-organizer, Dr. Shazia, she has to put a lot of effort in making the brochures, in designing the course program, and along with her colleagues, that is a marvelous her contribution. And I really appreciate to bring this course to this day, that we have more than 25 participants? 27 participants. And when she started the course, we were expecting that maybe not many, but we are happy that there are 27. Beyond this we can't take, because we want to teach you on-hand training in the laboratory. So we can't take hundreds. We were expecting only 25. So with this, thank you very much, and I wish you the enjoyable stay here, and look forward to your views about the course during your stay. Thank you very much.
Thank you so much, sir. Now may I invite the Honorable Chief Guest, Professor Dr. Fahim Aftab Saab, Dean Faculty, Life Sciences, University of the Punjab. So please come on the stage.
Thank you, Dr. Saga. Dear Professor Moaz, Director, Center of Excellence in Molecular Biology, Professor Idris, Professor Qasr Malik, Dr. Shazia Rafiq, one of the organizers of this event. And then I come to know that faculty members from other universities are here, so and participants, of course, and students and staff. It's really a privilege to be here and speaking with you, because as Professor Moaz was describing the outcome of the center, so I was just thinking that basically Center of Excellence in Molecular Biology is our pride as well. So we take things very seriously, and we normally hear something very nice from the center. Otherwise, you know that every institution is having so many problems. So I would like to welcome also the participants who are coming from faraway places, and I wish you all the very best, because I'm told, and as we have heard as well, that this technique is important, because I also do many in vitro experimentation. So once it is required to go into the field, then of course you can, but if you can handle thousands of plants, in the confines of a very small laboratory and in vitro setup, so this is an advantage. So I would say that the participants, I'm learning that some 27 of them are already here, and of course they will be attending this workshop. So you will be having that facility in hand to learn about cell culture. Of course, this has got its implications in biomedical research, drug discovery, regenerative medicine, and etc. And I think it goes even beyond. So what is desired from all of the participants is that they have put a lot of effort anyway in organizing this workshop for you, and you need to be curious, you need to be learning things that you can do to the best of your abilities, and you should interact as well. One thing Professor Saab mentioned, that enjoy and make friends as well. You can do that. Of course, you should develop your collaborations as well while you are working in some other environment, and you learn a lot. You know, whenever you are outside of your institution, you normally learn a lot from other people. But anyway, another thing that came to my mind was that you should give your feedback as well at the end. I think it will be part of the procedure, but you know, even sending a single email to Professor Saab and saying that this is what you learn, and this may include any bad experience as well. Any positive, feedback, if it is coming from you, that will be of great advantage. We can then see how to improve in future. But I'm sure that looking at the facilities, and I think you will be having an idea by now that you are perhaps in safe hands, you will be learning a lot. And one thing is that you need to be open-minded, discuss. I saw the program of this workshop, and I saw that there are time slots for discussion and Q&A. That is of course very important, but you will see that even during the lunch, during your stay here, you can have further discussions. So this is something very important, that you do things with an open mind and then try to see as if you can really implement that in your setup in the laboratory that you are working in. So that is of course important. So I think I don't have much to say because much has already been said, but I'm really delighted to be with the faculty and the trainees who are here. I wish all of them best of luck. And yes, the last point, which may not be the part of the discussion, but a solution-based approach was also, I think, mentioned by Professor Moaz earlier. So I think this is important for the younger generation to understand that in our times, mostly we were concerned with the basic research. And basic research is still important, remember, because without it, you cannot implement the practicability or the field research, as I would say. But the focus was on the basic research. Basic research gives you your MPhil and PhD as well, remember. But now the trends have changed. We have seen it's very important that you find a certain problem also on which you can work. So try to find out a problem where you can give a solution. This should be your approach, I think, for your future research endeavors after you earn your PhD. And you need to be equipped with those circumstances and those perhaps ideas which will be leading you towards that. So that is, I think, also very important these days. And as they say, that finding a problem is half of a solution sometimes. So, which is that if you are correct in your identification and saying that, okay, this is what is an issue that needs to be worked out, so then it means that you are on the right path. So develop these develop habits of reading too much about your subject. So this is, I'm saying to the students mostly, or the younger generation who are the participants. I'm sure that many senior faculty members are here, so they normally say the same, I think, in the classes and whenever they get that opportunity. So my best wishes to everyone for not only this course but beyond, and I'm sure that you'll be doing everything right, and then you will be having lots of memories about this workshop in the future. Thank you very much.
Thank you so much, sir, for your insightful words. Now may I request you to please come in front of the chairs and receive your souvenir from Dr. Maazur Rahman and the focal persons of the workshop. Please give a small hand. And when we hand over the souvenir to Dr. Saab, there should be a very big hand. Thank you so much, sir. It's time for group picture. Sir, आप लोग एक पहले स्टेज पे बना लें. आप चारों की स्टेज पे बना लें.
मैडम आप